At 3,769 meters beneath the ice of East Antarctica, a drill bit broke through the last few centimeters of a seal that had held for 15 million years. And the water came up to meet it. Not slowly, violently. The lake beneath was under such pressure that the moment the barrier failed, water surged hundreds of meters up the borehole like a geyser fired from inside the planet. And then, in the crushing cold of that shaft, it froze solid. It plugged the hole it had just escaped through. Nobody got to lower an instrument. Nobody got to look. The lake spat something up at the men who opened it, and then sealed itself again before they could react. That was February the 5th, 2012. The men were Russian. And when they came back the following season and cut out that frozen plug and carried it 3,000 kilometers north to a laboratory in St. Petersburg, the analysis came back with something that nobody on that team had prepared for. Genetic material. Thousands of distinct signatures. And one of them, cataloged as WON 2310, matched nothing. Not a species, not a genus, not a family. When they ran it against every organism ever sequenced on Earth, the closest relationship it had to any known living thing on this planet was less than 86%. To put that in perspective, you share more DNA than that with a mouse. Most people walk right past this story. It gets filed under polar research, somewhere between ice cores and penguin counts, and it dies there. But hidden inside it is something that shouldn't exist. And the reason you've never heard the ending is that there isn't one. The samples were disputed. The team fractured publicly. The drilling was suspended. And a second expedition, 1,000 kilometers away, and 6 years later, went down through a kilometer of ice into a different sealed lake and pulled up something even harder to explain. Bodies. Not microbes. Bodies. With legs still attached. We're going to work through all of it. The lake, the unmatched sequence, the retraction that wasn't quite a retraction, and the carcasses in the mud that made a veteran scientist assume his own team had made a mistake until he made them wash everything and do it again. Start with the thing itself, because the scale of it is what nobody communicates properly. Lake Vostok is not a pocket of meltwater. It is a body of liquid freshwater roughly the size of Lake Ontario, more than 250 kilometers long, over 800 meters deep in places. And it sits beneath almost 4 kilometers of solid ice, in the coldest place on the surface of the Earth. The station above it recorded minus 89.2 degrees Celsius in July of 1983, the lowest natural temperature ever measured on this planet. And directly underneath that reading, under a column of ice taller than most mountain ranges, is water that never freezes. It stays liquid for 2 reasons: geothermal heat rising from the rock below, and the sheer crushing weight of the ice above, which lowers the freezing point. The lake is trapped in a permanent balance between fire underneath and pressure on top. Now, the part that matters. How long has it been sealed? The estimates run from 15 million years to 25 million years of total isolation from the atmosphere. No sunlight, no oxygen exchange, no contact with the outside world of any kind. 15 million years ago, there were no modern humans, no Homo genus at all, and modern primates were still forming. Whatever went into that water before the lid closed has been down there, in absolute darkness, running its own private experiment for a span of time our species has no instinct for. For decades, scholars dismissed the possibility of anything living there. No light means no photosynthesis. No photosynthesis means no base of a food chain. The assumption was straightforward, and it was almost universal. Vostok would be sterile, a dead time capsule. Then modern science went down to check. The drilling started in 1990, and it was brutal work, possible only during the austral summer window between December and January. By 1998, the drill had reached 3,623 meters, and then it stopped, not because of mechanical failure, because of a problem that nobody had a clean answer to. To keep a borehole 4 kilometers deep from collapsing under the pressure of the ice around it, you have to fill it with fluid. The Russians were using a mix of kerosene and freon, which meant that sitting directly above the most isolated ecosystem on Earth was a column of industrial chemicals over 3 kilometers tall. Tens of tons of it, held in place by nothing but the last 130 meters of ice. Break through, and you dump all of it into a lake that had been cleaned since before our ancestors came down from the trees. International pressure shut the project down. Drilling stopped for 5 years while the argument played out. And the solution the Russian team eventually proposed in 2003 was genuinely elegant, and it is also the source of everything that went wrong afterward. They would not drill into the lake. They would drill toward it and stop the instant their sensors detected water just below. Then they would pull the bit out. The lake's own pressure, higher than the fluid column above it, would do the rest. Water would rush up into the borehole on its own, hundreds of meters, and freeze. Clean lake water, frozen into a plug, which they could then drill out the following year as ice. The lake would sample itself. And that is exactly what happened on February the 5th, 2012, at 3,769.3 meters. 22 years after the first bit turned. The plug came out the following season and went to St. Petersburg. And this is where the story stops being a triumph of engineering and becomes something else entirely. The analysis was led by Sergei Bulat at the Petersburg Nuclear Physics Institute, and the standard for this kind of work is savage, as it should be. You have to eliminate everything. Human skin bacteria from the people handling it, organisms living in the drilling fluid itself, dust particles in the clean room, contamination from the borehole walls. You strip out every sequence that could plausibly have come from anywhere except the lake, and you see what survives. Almost nothing survived, but not quite nothing. One phylotype came through the filter and could not be assigned to any known branch of the bacterial tree. Less than 86% similarity to anything cataloged. And here is the detail that keeps this alive. It clustered with a previously unidentified sequence that had been detected years earlier in accretion ice from the same site. 2 separate encounters with the same unknown thing. If you want to know what else came out of that borehole, subscribe, because this channel goes back into the sealed places that get quietly closed, and this one was closed fast. Because that announcement did not survive contact with the outside world. Broader analyses of the same material reported genetic signals from as many as 3,507 potential organisms, bacteria, fungi, and, most provocatively, eukaryotes, complex cells. Some sequences were reported as strikingly similar to those of common aquatic invertebrates. Here's where it gets strange. The challenge did not come from a rival institution. It came from inside the Russian team. In March of 2013, Vladimir Korolyov, who headed one of the laboratories involved in the analysis, went public and contradicted the result. His objection was specific, and it was devastating. The bacterium in question, he pointed out, could metabolize kerosene. Kerosene, the drilling fluid, the exact substance suspended in a 3-kilometer column directly above the sampling point. An organism that eats the thing you drilled with is not proof of alien biology in a sealed lake. It is the single most likely contaminant you could possibly find. And with that, one observation, the discovery of the century, became a laboratory error, and the story left the news cycle within days. But that explanation has a problem. It explains one sequence. It does not explain the earlier detection of a related unknown in accretion ice sampled before that borehole ever reached the water. And it does not resolve the deeper issue, which the researchers themselves acknowledged in the Philosophical Transactions of the Royal Society. With sample biomass that tiny, the work is only meaningful if you have a complete library of every possible contaminant to subtract. Negative controls sham extractions, human-associated bacteria, drilling fluid, dust microparticles. Nobody has that library. Nobody has ever had it. Which means the honest position on Lake Vostok is not that it was contamination and not that it was life. It is that the sample was ambiguous and no one has gone back to settle it. There are no plans to drill that portion of the lake again. In the absence of putting a camera down there, the contamination problem is permanent. The most isolated body of water on Earth was opened once, gave an answer nobody could verify, and was left alone. That is the first lake. Now we get to the one nobody expected to produce anything at all. Roughly 1,000 kilometers away, beneath the Whillans ice plain in West Antarctica, sits Subglacial Lake Mercer. It covers around 160 square kilometers, twice the area of Manhattan, and it is shallow, 10 to 15 meters deep. It was found by accident. Helen Fricker, a glaciologist at Scripps, spotted it in satellite altimeter data in 2007 while doing something else entirely, watching the ice surface above it rise and fall as water moved underneath. Expectations were low, and they were evidence-based. In 2013, an American team had drilled into nearby Lake Whillans, the first clean access to an Antarctic subglacial lake, and found microbes, only microbes. That result set the ceiling. Sealed subglacial lakes host simple life and nothing more. On December the 26th, 2018, the SALSA project melted a borehole roughly 40 centimeters across through 1,087 meters of ice using a clean hot water drill, and held it open for about 10 days. 50 people, water samples, ice properties, current measurements, and the longest sediment core ever taken from a subglacial lake. The discovery did not come from the core. It came from mud. David Harwood, a micropaleontologist from the University of Nebraska-Lincoln, was cleaning lake sediment off an instrument in a shipping container fitted out as a field lab, and decided to look at the residue under a microscope. He was hunting diatoms, fossil algae, routine work. What he found, instead, was a shell, a shrimp-like crustacean. Then, a tardigrade, the 8-legged microanimal famous for surviving conditions that kill everything else. And its legs were still attached. Its carapace was speckled and discolored, he said, like an old leaf that had been lying on the ground for a season. Then another fragment, this one amber-colored, still bristling with fine hairs. These were animals between 1/10 of a millimeter and 1.5 millimeters across, but animals in a lake sealed under 1 kilometer of ice where the established science said there should be nothing but bacteria. John Priscu, SALSA's chief scientist, did not believe it. His immediate assumption was that his own team had contaminated the samples, so he ordered everything cleaned again, thoroughly, and had them resample. The new cores came back with more crustacean shells. And here is the detail that turns this from a curiosity into a genuine open question. The specimens do not appear to be lake creatures, and they do not appear to be ocean creatures. The tardigrade and some of the other remains look like land dwellers, terrestrial animals, recovered from beneath 1 kilometer of ice in a lake that has been cut off for millennia, in a landscape where the nearest exposed ground is the Transantarctic Mountains, hundreds of kilometers away. The mud around them also held remnants of algae from a period when Antarctica was dramatically warmer. And Priscu's working explanation for the microbial life is that an ocean covered the area roughly 1 million years ago. That accounts for marine material. It does not account for land animals. As of the reporting, how they got there remains unresolved. Now, does any of this prove there is something alive and unknown under the Antarctic ice? No. And you should be suspicious of anyone who tells you it does. The Vostok sequence has a plausible, mundane explanation, and a member of the team that produced it said so out loud. Kerosene metabolizing bacteria in a borehole full of kerosene is the least exotic finding imaginable. The Mercer carcasses are dead, and they may be very old, blown or washed into a basin during a warmer period, and buried when the ice returned. Neither result requires anything that overturns biology. But look at what the grounded version actually says. It says that beneath the largest ice sheet on Earth, there is a hydrological system of hundreds of interconnected lakes, holding liquid water that has been isolated for geological spans of time. And that, when we have gone down into two of them, both times we found more than the models predicted. Willens gave microbes when the expectation was sterility. Mercer gave animal remains when the expectation was microbes. Every time the drill goes deeper, the ceiling moves. And it says something else. Antarctica was not always this. The algae in that mud are real. The continent underneath the ice held forests and open water, and everything that lived there was buried, not erased. That is the frame that matters. This is not a dead continent with ice on top. It is a preserved one. If the ambiguous signal in Vostok is ever confirmed rather than dismissed, it would mean an ecosystem ran for 15 to 25 million years with no sunlight and no contact with the surface, and evolved down a branch that separated from everything else we know before our lineage existed. That is not a footnote. That is a second experiment in life on this planet, running in parallel in the dark the entire time. It would also mean something for everywhere else. Europa has a global ocean under an ice crust. Enceladus is venting liquid water into space through its shell. The reason NASA watched Vostok so closely is that the problem is identical, only easier. If we cannot resolve whether something is alive under 4 kilometers of our own ice without contaminating the answer with our own equipment, we are not ready to ask the question on a moon 600 million kilometers away. And the honest ending is that we did not resolve it. We opened the oldest sealed water on Earth once, got a reading nobody could confirm, argued about it in public, and walked away. The lake is still there. 250 kilometers of black water under the coldest air on the planet. Still liquid, still pressurized, still sealed, except for one narrow shaft that froze itself shut 14 years ago. The plug is still in the borehole. The unmatched sequence is still in a database in St. Petersburg, with no relatives. Nobody has sent a camera down. Nobody has permits to go back. Whatever answered that drill in 2012 is still down there in the dark, and it has been waiting a lot longer than we have. If you want to know what else is buried under the ice, watch the next investigation on screen right now.